Good afternoon or good morning, everyone, and welcome to our webinar. My name is Michael, and I will be your host today. I've been with Cube Biotech for about 7.5 years, and I have had the opportunity to see our membrane protein technologies evolve from early concepts to the automation-ready workflow we are discussing today. We have said for years that we want to make membrane protein purification easier. Today, we will show you the data behind that statement. In this webinar, we will present the real performance data on automated membrane protein workflows using our new launch product, Plate XMP, including reproducible purification results from 8 GPCR targets and implementing insights from active drug discovery programs. I'm very pleased to host the sessions together with two outstanding experts. First, we have Philipp Harnisch. He is head of lab at Cube Biotech. Philipp holds a PhD from Heinrich Heine University Düsseldorf and brings extensive expertise in membrane protein expression purification. Before joining Cube, he primarily worked with classical detergent-based workflows. After being introduced to polymer-based stabilization technologies here at Cube, he quickly recognized the ability to better preserve native lipid environments and protein stability compared to traditional detergent systems. Today, he leads our lab team in translating that potential into automated workflows. We are also joined by Valentina Shchedrina, a senior scientist at Orogen Therapeutics. Valentina has strong background in biochemistry and protein science with previous research positions at Harvard Medical School and the University of Nebraska. At Orogen, she focuses on GPCR drug discovery and screening platforms and brings hands-on experience implementing automated membrane protein purification workflows in active drug discovery programs. And also, before we begin, please feel free to submit any questions using the chat function. We will collect all questions and address them at the end at the Q&A session. So you can just write every time in the chat. And with that, let's get started. Philipp, the stage is yours.
Thank you very much. Do you pull up my presentation or should I do that?
No, I should be shared now. Yeah, okay.
Okay, perfect. Good. You can all see my pointer, I hope. And then we are ready to go. Yeah, thank you very much, Michael, for this kind introduction. And thank you all of you for attending this webinar. I'm going to give the introduction to Plate XMP today before Valentina will then show her actually pretty damn amazing data. So today we're going to talk about Plate XMP. But first of all, what is Cube and what are we doing? So Cube is a CRO and a product business, and we provide innovation services and products for especially membrane protein research. In our lab, we have purified more than 400 unique proteins and more than 300 membrane proteins. And what we love to do is we love to go full length, and we love to not introduce any modifications into the protein. So we like to work with the full length protein. We do it quite successfully. So our actual success data at the moment is more than 95%. So there's a few targets which are very hard to get, but we're going to get them all. We are founded in 2012, and right now we are a team of more than 100 people, and we're also very strong in patenting our technology. So we have more than 50 active patents. Valentina, can you maybe switch off your pointer because I can see it in my presentation? Thank you. Okay. So why do we like to work with membrane proteins? Yeah, first of all, because membrane proteins are amazing, and second of all, because membrane proteins are very hard to work with, and it's a great challenge. So if you take a membrane protein out of its natural lipid environment, it doesn't like that so much, and if you do not find a way to stabilize it, it's going to just be completely inactive, it's going to aggregate, and you're not going to have a good time. So over the past 50 years, there have been a lot of different approaches to stabilize membrane proteins. And the most common way has been or still is detergents. So those detergents, they can form a micelle around the hydrophobic core of the membrane proteins and stabilize it this way. But the issue is you're going to wash away nearly all, sometimes also all of the lipids which are stabilizing the membrane protein. And this can lead to misfolding and misconformation. So people came up with a lot of different techniques to stabilize those membrane proteins. They were just recombinantly expressing the soluble regions, or thermostabilizing by protein engineering, by introducing mutations, or then after extracting it with detergents, putting it back into an MSP nanodisc, which has lipids, but those are, it's never the native lipid environment because it's hard to guess. So people were experimenting around with a bit of different lipids, concentrations and lipid conformations, but all of this, to put it together, the function interstability is often compromised. You have therefore a high risk of projects with a long timeline, sometimes more than a decade for some proteins. This then comes with very high R&D costs. You often end up with non-native results, and the best example is like there was no full-length GPCR really unmodified which ever could be stabilized and solubilized in detergents properly. So that's an issue. And the scientific community now more than ever found out that the protein-lipid interaction is really crucial. There have been so many examples where great scientists can show or do show that the protein and the lipids, they are a unit, they work together, and without a native lipid environment, it is often very hard for the protein to do its proper function. And this is also the reason why membrane proteins are still a bottleneck, even though 60 to 75% of the top-selling drug targets in 2025 were membrane proteins. All in green, all the drugs in green, they were built up on membrane proteins, which is actually quite impressive. So due to all these factors, you have limited scale in pharma and in academia, and it makes it very hard to work with these membrane proteins. So yeah, but who would we be if we wouldn't have a solution for this? And this is called our native MP platform. The native MP platform builds on amphiphilic copolymers, and those amphiphilic copolymers, they can find their way into any biological membrane, they destabilize the membrane, and then they cut out native nanodisc. Means you have a belt which looks a bit like an MSP, but it's not derived on lipoprotein 1, but on the synthetic copolymer, and it can retain the native lipid environment. This is very important. Native lipid environment of the protein gets retained with the copolymer technology, meaning you do not need any detergents whatsoever anymore, and you can very easily solubilize and stabilize a membrane protein one step in a nanodisc-like environment. This keeps the function preserved because we preserve the natural environment. For the membrane protein, not much really changes. It is detergent-free. We saw that this technology provides us with a never-seen stability before. We have examples where we had stability for more than several weeks at room temperature. You can also perform your assays at body temperature. So 37 degrees is not an issue anymore compared to detergent, where this was most of the times nearly impossible. And we therefore have also a lower risk and faster timelines. And now we finally also can work with full-length GPCRs. Okay, we have a huge portfolio at the moment at Cube Biotech. So we have 32 different copolymers. But for the innovation we're going to show you today, we concentrate on the Cubipol backbone. This is our in-house derived product. So our chemists, they developed this in-house, and they looked around in all the different copolymer backbones and found the best described or best working site modifications of this backbone and introduced them into the Cubipol. So this is the first backbone now where you have all the different site modifications you can find throughout the public research combined in one backbone. And this makes it very powerful. And this is why we chose for this product to go with Cubipol. Yeah, but what's actually the issue? So if you work with classical detergents, the isolation process takes minimum 2 days, sometimes 3 days, 4 days. This is not so different with copolymers, but with our product today, you're going to see how this can change. So classically, you lyse your cells, you centrifuge your membrane down, you solubilize the membrane with detergent, you purify it, and then you end up with eluates. And very often, the issues are that purity and the yield can be not so good. So as you can see here, we made a screening with 5 different detergents. We did this in my lab, and you can see we have lots of impurities. We also have a very heterogeneous sample. So the detergents, they sometimes destabilize the protein. We get some aggregates. And even for those samples which were looking good, the protein was not very temperature stable. So at 40 degrees, we already see like an inflection point in the cumulative radius, meaning it aggregates. Okay, but if we do the exact same thing with copolymers. These are the exact same proteins from the exact same pellet. Just with copolymers, we can see way nicer bands in SDS page. We have very homogeneous distribution in DLS. And what's most impressive is we have an increase in temperature stability of more than 20 degrees. And this is outstanding. This is something which is really rather quite unbelievable. So this is why we like to swear on these copolymers and why they are so amazing. Okay, but the process, as I said, is still pretty much slow. We have to screen by hand. We provide them already in a lyophilized state. They are pre-buffered. This makes it already very easy to use, but still it's iterative, it's slow, and you have a low throughput. So we were thinking for the last year how to come up with a solution to make it easier for you, and of course to make it also easier for us in the lab. And the most easy thing is if you can automate everything. So we went out there and we talked to different companies, and we found 2 collaboration partners for the beginning, 2 liquid handling systems, the SciBioPhilix from Analyticena and the Assist Plus from Integra, to put out an automation platform, to make it fast, to make it reproducible, to make it high throughput. And the product, which ended up being the product we're going to show you today, this is this plate, this plate XMP. In this plate, you find everything you're going to need for the purification. We have the copolymers, we have an empty column for your lysate, so you only need to lyse your cells, spin on the cell debris, and fill it in there. You have dried magnetic beads. I'm going to later show you how this looks, and you have all the buffers you're going to need for the purification. So you're going to need only water, and you're only going to need your cell lysate, and that's it. You have 8 different cubipoles in the plate. Means you can have fast parallel processing. You have identical handling across all these conditions, a very high reproducibility since the liquid handling robot is doing this for you. This brings me to this slide where we say it's a walk-away workflow because you really just have to press the play button. You walk away for 2 hours. You have a coffee. You have a chat with your colleagues. You do another experiment if you're good. And then this is already all you're going to need. So there's fully standardized steps. And the best thing here is we brought it to 3 different tagging systems. So for the row tag, for the flag tag, or for the strap tag, with strap tag TNXT MacBeats in there. And then you end up with purified eluids in just 2 hours. Okay, so how is this possible? How can we dry everything? Our chemists, they found pretty amazing technology, as far as I can judge, to dry the MacBeats. So they create a film with MacBeats, which can be then resuspended very easily by the liquid handling robot. They lyophilize the copolymers directly into the plate, and they also lyophilize all the buffers you're going to need on the plate. So the plate arrives at your place, completely dried. You can just store it in the fridge. If you want to start, you rip off the seal, you fill in your lysate, you press play, and that's all you have to do. We've done 2 application notes with those companies, Analyticena and Integra. And there we show that this works with GPCRs, and even more impressively than we could ever show, this Valentina is going to show you in a minute. We used single pass proteins and other multiple different membrane proteins, and even got a structure with P2X4 right from the plate. So if everything goes smoothly, this is what is even possible. So now with this plate and our native MP technology, you can detergent-free extract native lipids with your copolymer. You can preserve function and structure. You can use this wonderful 96 well plate, easy to use, like everybody can purify membrane proteins with this, and it comes with lyophilized reagents. And if you're looking for downstream applications, also this is what we tested over the last 5 years. I would say there's really no limit to what's possible. So we have done cryo-EM. We saw in collaboration with the Chernyshev lab that X-ray crystallography or cubic phase crystallization is also possible now. You can use it for screening and validation. You can use SBR, the BLI. You can use spectral shift for it. You can create antibodies, or you do small molecule drug development via ASMS or dead screen. This is all what we have shown. And with this, I would like to thank you very much and hand over to Valentina. Michael, you can now pull up the presentation of her, and then she will really show this wonderful.
Yep. Thank you very much, Philip, for explaining the concept.
Thank you, Philip.
As I mentioned, ask questions. We will answer them after both talks. Okay. Can you do it, Philip? It does not work with me, unfortunately.
Of course, you go.
Hello. My name is Valentina Shedrina. I would like to thank Michael for nice introduction. I'm a senior scientist at Orogen Therapeutics, and I also would like to thank Philip for introduction of copolymers. I have one slide, but of course it's better to hear about copolymers from person who produce and sell them. So today I will talk about rapid and efficient robotic purification of GPCRs for heat discovery. My talk has 5 parts. Introduction, then I will compare a manual and robotic purification of 8 GPCRs, and to solubilize these GPCRs, I use detergent or copolymers. And in my last slide, I will share my vision how we will purify GPCRs in future based on the knowledge that they got from robotic purification of GPCRs using copolymers that I will present in part 2 and part 3. And finally, I will give conclusions. Philip already described copolymers, but I would like to here mention that GPCR that solubilized and extracted using detergent, I will image on my slides with blue detergent on my SDS pages, and GPCR that is solubilized and extracted using copolymers, I will show on my slides with green copolymer. And I took this slide from Cube website. Cube introduced us copolymers in March 2025. Michael came to our company and he described the screening kits. We used the kits. They worked perfectly. We were able to screen our GPCRs and find optimal copolymer for manual purification. And recently, in December 2025, Jan Kubicek and Barbara Mertens visit us again, and they suggested us to try their new product, Plate X MP. It's a 96 well plates that Philip already mentioned. They have 8 copolymers loaded here, and also Cube allows us to interact with their partner, Sai from Analytic Jena. Brian Sates came to our lab, installed their robot, and also I think he one who developed software to run this robot. Philip previously mentioned that purification lasts for 2 hours. I confirm it. Basically, you can get your GPCR in 2 hours if you load cell lysate here. And I spent 2 hours to prepare cell lysate. So total purification, excluding SDS page, lasted for 4 hours for me. And 2 hours during this purification, I did nothing. So it's very easy and convenient way to purify GPCRs. And it's very effective way. I will show it later. So we only used our cells. All other reagent and device was provided to us. And I'm very grateful for this opportunity because at the end, we figure out how we will purify GPCRs in future. So on this slide, you see purification of 58 kilodalton GPCR. This purification was made manually, and I used detergent. This is my cells. Detergent molecules. And this is samples from robotic purification. I have 7 copolymers because in this specific case, copolymer number didn't work properly. I pulled all of these samples, and this is a resulting pulled sample that was used for DEL screening, and this sample was also used for DEL screening. As you can see, purity of both samples purified manually or using robot is compatible. However, yield is significantly higher in case of robotic purification, and I spend less time to finish the process and to get the amount that is enough for DEL screening. I got 70 microgram of GPCR from 22 grams of insect cells. And in case of manual purification, I got 180 microgram from 132 grams of insect cells, and less time. And I will have the same conclusion for all my purifications. In case of robotic purification, my yield was higher, and I spend less time to get compatible or required for the screening amount of GPCR. This is another GPCR. In this case, GPCR was purified manually using detergent, and I have some cleavage here that was confirmed by Western blot. In case of robotic purification, I didn't see any cleavage, and after pulling all my samples, I got GPCR with purity compatible that I got through manual purification, except I have cleavage here. And again, yield was higher in case of robotic purification, and I spend less time to accumulate the amount of GPCR that is needed for DEL screening. Both these samples were used for DEL screening. One more purification of 81 kilodalton GPCR. In this case, I used copolymers for purification manually, and also cube plate allows only copolymer-based purification. Purity is compatible. Yield is higher in case of robotic purification, and I need less time to get a compatible amount of GPCR, 54 microgram here from 180 gram of insect cells, and 75 microgram of GPCR from 5 gram of insect cells. One more GPCR, 101 kilodalton GPCR. It is supposed to be dimer. This is the result of manual purification. There I solubilized GPCR using SMA copolymer. We found that this copolymer is optimal for this specific GPCR using cube screening kit, because this saw better image on SDS page, and also amount was higher for specifically this copolymer. In case of robotic purification, I also was satisfied with purity and amount of protein, and I think it's compatible quality of for robotic purification and manual purification. However, yield is higher in case of robotic purification, and I spend less time to get the amount that is required for DEL screening. We did screening for this specific GPCR, but amount you can compare. It's 75 microgram from 7 gram of insect cells here, and 50 microgram from 145 gram of insect cells. But at the first step of purification, I do membrane purification. This is probably why I lose a lot of protein. Then I purify proteins manually. But even ignoring amount of the protein, this process is significantly faster. It's 2 hours purification, and you don't need to do anything. This is one more GPCR, 83 kilodalton GPCR. Also, purity is compatible for manual and robotic purification. In both cases, we used copolymers. This is cubipole copolymers, and this is SMA copolymer that we selected during screening process. It gives us optimal purity. Yield is higher in case of robotic purification, and one more time, I spend less time to get compatible amount of protein in case of robotic purification. Another GPCR, 71 kilodalton GPCR. Robotic purification gave me, in my opinion, dirty samples, but I need to mention that this is cubipole copolymers, and they were not optimal for this specific GPCR because we did screening, and I saw that cubipole copolymers didn't work well for this specific GPCR. This is why I selected SMA copolymer for purification. But anyway, yield was higher in case of robotic purification, and to get equal amount of protein, I will need to spend less time in case of robotic purification. One more GPCR, 69 kilodalton GPCR. Manual purification, robotic purification, in both cases we used copolymers. Purity is compatible for manual purification and copolymers 1, 2, 5, 6, 7. I have some cleavage here, or I don't know what is it, and these two copolymers didn't work well for this specific GPCR. However, even if I pulled fractions, samples from line 1, 2, 6, 7, I will get higher yield if compared with manual purification. So here I have 66 microgram for 5 gram of insect cells, and here I have 140 microgram from 136 gram of insect cells. So in my opinion, quality is compatible, yield is higher, and time, I need less time in case of robotic purification. And this is my last slide. Here I would like to share my insight, my vision, how I will work with our GPCRs in future. In this case, GPCR was expressed in mammalian cells using transient transfection with BACMAM construct. And from one purification, I got 83 microgram of protein from 6.76 gram of mammalian cells that were grown in approximately 500 milliliters of media. So this type of experiment will save me time and money because I will not need to produce liters of insect cells, and I will not need to produce baculoviruses, and it will save me a lot of time. And purification total time without SDS page lasted for 4 hours from sample preparation to the moment then I loaded my samples on SDS page. So in future, I will do transient expression of GPCRs in mammalian cells and use our robot to purify GPCRs. And at the beginning, I will do screening actually using Cube kits. This is how I see our future purifications. And to summarize, Cube workflow, plate X and PTM plus automation is a revolutionary step for GPCR purification, delivering exceptional yields up to 10 times higher in comparison with manual purification across multiple GPCRs. I presented 8. Robotic purification of 8 GPCRs was faster, hours instead of days, and cost-effective, up to 20 times less biomass needed to purify the same amount of GPCR. The GPCRs purified using copolymers were more stable 2 weeks at 4. I tested only 2 weeks. 2 weeks at 4 degrees. Then GPCRs purified using detergent-based approach, several days. Actually, I would like to mention that I lost one GPCR during purification when I did manual purification. So GPCRs not stable proteins for sure. The protocol was very simple to work with for a person who has never used automation before, me. So I never used robots before, but Cube was able to develop workflow that works very effective and very fast, and I love it. And finally, I would like to thank people who helped us to test this technology. It's Michael Rees, Jan Kubicek, and Barbara Ertons. And also I would like to thank Brian Sales, who installed a robot in our lab and also answered my questions. And people who generated baculoviruses that were used for expression of GPCRs. And also I would like to thank the audience for listening to this, and also Cube, who invited me to share this data. That's it. Questions, if somebody had them.
Yeah, thank you very much, Valentina. So we have actually perhaps too much questions for half an hour discussion. So we also planned enough time, but the questions are flowing. So I will display them. I will go from the first one to the last one, and really try to answer as much as we can. Okay, the first question is from Katharina Zau. Is this single-use plate? So Philip, I think you can answer this best. Or also
yes, yes, so we have the plate is pre-formatted with all the buffers, with the MAC beads and the copolymers. The copolymers are going to be used up after you use the plate. The buffers are going to be used up after you use the plate, and the MAC beads, you can, if you want, you can pipette them out and clean them and use them again, but the plate itself is single use.
Then we have another question from Miguel. Would it be possible to use the plate without liquid handler, but using multi-channel pipette?
I don't know. I think the robot is a magical device. I think my high yield somehow comes from robot, not from pipettes. I don't know. I don't know answer. So.
I agree with you. So if you have a very, very strong arm and a lot of stamina, you can pipe it thousands of times, but it's a bit like you can also walk to vacation or take the car. That's the liquid handlers to take the car, and pipetting behind us to walk. So if you don't have a liquid handling robot, I would still advise to use the normal screening kit and maybe save a little bit up for the liquid handler to then use this.
Next question from Sheila. Hi, thank you for the presentation. Can we use the plate without the Felix and the liquid handler? Okay, this is the same question, basically. You can do it. It will be probably painful. It's advised then to buy the reagents by themselves and do it manually. But as Valentina also mentioned, the advantage with the higher yield comes probably from the robot.
I'm pretty sure it comes from the robot, yes.
Then we have another question from Lucas. Thank you very much. So that's in your initial protocol for the 30 polymer screening kit. After solubilization, the soluble proteins are separated from insoluble parts by ultracentrifugation. Also, it was advised to dilution the polymer binding to magnetic beads. Now this is also skipped, or does it only work for the 8 polymers in the MP plates, but not for all 30 from the screening kit?
Okay, so this still holds up our recommendation if you do it by hand, because we have proven it to work better if you do it by hand. But nevertheless, with the robot, it's not necessary anymore. So you can just pipette crude lysate in there. You can even, if you add DNAs before, use whole cells in there. You don't need to dilute it anymore. The pipetting power and the mixing power of the robot is so good that you have such a good mixing of the MAC beads with the lysate that it's going to be so super powerful. This is what we saw, and what also Valentina can agree, I hope.
Yes, I agree. I don't know how, I actually didn't believe. Then I saw this yield, I didn't believe it. And then I decided to use, you gave us all these plates just to test them, but I decided to produce sample for Dell screening using these plates. But yeah, I was very impressed with yield. Yield was fantastic.
Also, these answers show again that the magic comes really from the robot combined with the polymer technology. Is there a suggested protocol for the lysate, and is it compatible with both bacteria and yeast cells?
Yes, as long as you use like open cells, so a lysate, you can use it with any kind of cells. The polymers cannot penetrate gram-positive bacteria because of the peptidoglycan layer, but as long as you provide a lysate, it's going to work with all biological membranes.
Then we have again from Miguel, how is the FLAG protein eluted with low pH or competing peptide?
Everything in our plate. Oh, sorry, Valentina, please.
I never used FLAG. I always used RHO. I'm a huge fan of RHO magnetic beads. For all my GPCRs, I used only RHO. So I don't know FLAG.
Yeah. So for all of our plates, we always use the peptide. So for Rho, it's the peptide. For FLAG, it's the peptide. And for Streptactin XT, it's biotin, 50 millimolar solution. And that's also my recommendation. Low pH, and membrane proteins. And I don't really like this. So the peptide is better.
Then another question, also from Miguel. Our standard HEC 203 cells are lysed with mild detergent. Can such lysate be used in your system? Would any additional detergent interfere?
No, don't do it. So you can use the whole cell if you want, or you can just like open the cells via sonication or French press or whatever way you want, but don't use detergents. It defeats the whole purpose of the whole copolymer story. Yeah.
I agree, yeah.
Again, from Lucas, a question. Do you always screen the plates at room temperature or also at 4 degrees? Most pipetting robots do not work at 4 degrees. Or are your native nullers generally stable at room temperature? So yeah, Lucas, that's a great question. And we do everything at room temperature. Hopefully Valentina did also. But we also especially see if we use it at room temperature, we get way higher yield. Yeah. And as also Valentina also teased, these are more stable and they're also stable at room temperature.
I would like to add that once I left samples in the robot and they were at room temperature during the night, and then I analyzed them in the morning, they were fine on SDS page. I didn't make any activity testing, but on SDS page, GPCR was just fine. So room temperature.
Yeah, from Daniel, will this work for bacterial expression system, gram-positive and negative?
Yes, I think we've already answered this. It will work as long as you provide a lysate.
Okay, then we have a question from Nikolai. This is for you, Valentina. Can you comment dimer versus monomer on the 101 kDa GPCR case, active state and so on?
I don't know, because we don't measure any activity. We use our GPCRs for Dell screening. And the only type of assay that we do is a Dell screening. And currently, I don't have access to data that we obtain using Dell screening. So I don't know, is it active or not? But on SDS page, I can judge only purity, no activity. We don't make any activity measurements. And it's really hard to do activity measurements for GPCRs. I don't know, especially then protein is purified. So I cannot answer.
Okay, then we have another follow-up question from Nikolai on this is regarding the case where you observe the cleavage. Is it cleavage from the GPCR? Some co-purified partner SI did not see the expected shift in size of the GPCR due to the truncation. It looked similar size in both cases, but the degradation was 25 kDa.
Cleavage. I had cleavage on two GPCRs. One GPCR I confirmed by Western blot. I did Western blot with, for example, Rechotac, because my GPCRs also has Histac, Flactac. But another GPCR cleavage I saw in robotic purification, and I don't know what is it. I didn't analyze sample, and probably it's co-purified partner, but I didn't analyze this sample. The goal of experiment was just to see how this robot works. And for me, it was tiny amount of cells that I used for robotic purification. So the goal was just to see that a robot works. What product I get as a degradation, I don't know. I was not interested even to study. If I need to purify this GPCR using robot, I will just choose another copolymers that give me clear picture without degradation or cleavage or just additional partner, whatever.
Yeah, it's always depending on what you look. Thank you. What about optimizing buffers for stability regarding cryo-EM or initial solubilization? Is this done for good hits, or did you get a structure just from one run? Does it make sense to try the full screen for different buffers?
Yes, of course. Okay, that's a very good question. So, of course, if you want to develop a product for the broad majority of all membrane proteins, you have to find a way to have a buffer which works for most of proteins. And this is the buffer we chose. This is 150 millimolar sodium chloride, 20 millimolar HEPAs, and a pH of 7.5. And we see that this buffer works exceptionally good through the bank of a lot of membrane proteins. So the structure we actually got from P2X4 from one plate run was in this very same buffer, and it was enough to check different of the Kubi-pool variants to find the one which gives us amazing results. Of course, this was a very lucky shot, and I will not promise you get a structure from one run with a protein you try for the first time. So that's the starting point. It's a starting point to identify the best copolymer for you. You can do some biophysical, some biochemical analysis of your protein, find the best fit, and then go into upscale, try different buffers out. And this already gives you a huge advantage on your way to success.
Thank you. Next question from Dan. What expression scales are typically for solubilizing with these polymer-based nanodiscs?
For robot, it's 5 gram. 5 gram of insect or mammalian cells, which was cube advised. So 5 gram you can get from probably, I don't know, 500 milliliters of insect cells. It depends actually on type of insect cells. SF9 cells give less biomass in comparison with SF21. So it's a tiny amount of biomass for robot.
Yes, exactly. We use 300 milliliter of expression culture, and this gives us around about 5 grams. Exactly what Valentina said. So it's not much.
Good. Next question from Mark. Sorry that I have to rush because we have so many questions and I really want to answer them. Can you comment on your current protocols surrounding polymer purified protein on ICS pages? Poly removal still recommended?
No, I just have sample, add SDS page buffer and run SDS page buffer. I do not remove anything. And I don't think it's polymer specific, no. It's probably a question about charge of the polymer, right? I didn't see any artifacts on 30 copolymers that Cube suggested in the screening kit. So all my SDS pages were fine. I just added sample buffer. I didn't remove any copolymer. No, it's fine. They're fine.
Yeah, we also don't remove any polymer anymore. I want to keep it as easy and as simple as anyhow possible. And I want to have high throughput in the lab. So we just heat the sample at 46 degrees for 30 minutes. After addition of the SDS buffer, then put it on a gel and run it. That's it.
I don't heat it because I tested heated samples and unheated samples, and I didn't see difference for several GPCRs. So I don't heat even samples. I just add sample buffer and load it, and it's fine.
That's good for you. Sometimes you then have the sample stuck in the pocket, and if you heat it too long, you have it again stuck in the pocket. So sometimes it's a bit of a sweet spot, but that's individual. Yeah.
GPCRs or transmembrane proteins?
Transmembrane protein. So it's not only GPCRs. It's like, since we have all these different kinds of proteins, we have standard procedure. But if this works for you, it's amazing. It's even faster.
Probably for. Okay, sorry.
Next question we have from Stefan. What is the cell lysate? Purified membrane fraction or complete cell lysate? And how much is loaded per plate X, and what is MP yield per plate?
Okay, so the lysate is just we sonicate our cells, we spin down the crude rest of whatever is there at 9,000 G, and then we use this for the plate, and you can use 1.8 milliliter per well.
I confirm it. I was actually in shock in the protocol. I didn't believe it because, you need just buffer that has protease inhibitors, and you need to syndicate and spin down cells, and that's it. And then you load tiny amount per well, and it works fine. I thought I will have cleavage, but no, in most cases I have nothing, no cleavage. It works very well. And they didn't pay me. They didn't pay me. I'm not the agent. No, I just got a device for testing.
What are the factors that make it that automated purification leads to much higher yields than manual purification? I really like this question, and we also debate a lot about it. Philip, what do you think?
My personal belief is that the high mixing capacity of the MEC beads in the lysate itself, so this high mixing frequency, makes all the difference. We see this if we use a Kingfisher Flex. It even works better than with a pipetting robot, because I believe the mixing is the key. If you add MEC beads to your lysate and you just put it on a roller, it's not going to see much of the lysate in its time, because the MEC beads, they sink down and they don't mix so well. But if you force them to really heavily mix, this is what it makes the difference. At least that's my personal belief.
Okay, then the next question, which thanks, Guy. What can we use the Kingfisher Apex system for this workflow? So I can also answer this. Yes, you can, but not with the plate that was shown. The setup would be different, and especially the setup that we have ready for the Kingfisher is also meant for like screening 2 or 3 or even 6 membrane proteins. But this is possible to use with the Kingfisher Apex. If you want to know how, we just reach out to you afterwards and tell you this will be a product for, 2 to 3 months. Then perhaps you like this. Would the SMALPs survive a tween washing buffer as used in the lasers?
Yeah, they survive this buffer.
Okay.
I don't know.
I think I take now the last question. I'm sorry, it was too much. But I like also these questions. Hi, do you also use robot in a plate for large-scale purification? I agree the yield is great and the faster time help the GPCRs stay happy. However, it would be insufficient for cheaper experiments like cryo-EM. What are your thoughts on that?
I disagree. It is not insufficient for experiments like cryo-EM. For cryo-EM, you don't even need such a high concentration anymore. With polymers, like 1 mg per mil can be absolutely sufficient. And if you have a good expression, and it always ends up with a good expression, then you're going to end up with enough to even start doing cryo work. If you want to go into upscaling or use higher amounts, plate XMP is not the tool for this, but we're going to come out with a product in a couple of months, which is the tool for this. So just wait a little bit longer, and then we also got you covered on this side.
I also would like to add, because protein is stable. I tested one GPCR, it was stable for 2 weeks. Because protein too stable, you can do one by one purification and accumulate the amount of GPCR that you need. This is what I did for DEL screening. I did free purification, accumulated GPCR that is required for DEL, and give it to our DEL screening team.
You're right, this is absolutely also an option.
Okay, then I would like to close the session. So there's some questions open. Don't worry. I will send it to our experts, Philip and Valentina, and they will answer you shortly, perhaps in 1 week. Yeah, give us a little bit of time. So thank you really very much for your also engagement. This was very good. And we are very happy to showcase with you. Thank you again also, Philip and Valentina. And yeah, wishing you a good day ahead. Bye-bye.
Bye-bye. Have a great day.